speaker is Oleg Simakov from the University of Vienna, Austria. He is going to present his work entitled Syntenic Transitions in Metazoan Genomes and their Regulatory Impact. So I think he's already sharing the screen, so that's good.
Yeah, but we cannot hear him, right?
No.
Let me see if I can find him. One more sec. Someone knows an easy way of doing this?
No. I have it rear in my screen, Oleg, but I cannot mute him.
Someone saying you can search participants.
Mm-hmm.
Participants, okay, yeah, there we go. Thank you. Ask to unmute, there we go. Thank you guys for the tip.
Yeah, great. Hello, can you hear me?
Yes.
Great, yeah, awesome. Thank you to the organizers for having me here and allowing us to present some of our work that we've been doing recently. I would like to start with more like a general question about what is synteny and how this relates to potential regulatory transitions. We want to understand basically the question, why are animal genomes organized in the way that we see that they are organized? And this observation that there's some linkage between genes that is preserved across many species has been originally observed when comparing chromosomes of closely related Drosophila species and looking at the karyotype of karyotype conservation between different arms or whole chromosomes of those species. And this is how the term synteny, or genes that are on the same ribbon, was coined, and those chromosomal arms that were homologous were called Mala elements. But nowadays, with more genomic sequences and more kind of insights into genomics, comparative genomics, we know a little bit more about synteny, and particularly we now know that that there are two types of synteny. So this, on one hand, this macrosynteny. These are the small 3 to 10 gene clusters. There are many of them in animal genome conserved between multiple animals. However, only a few of them have been really studied functionally. For example, the Hox cluster, maybe there's some evidence for the Wind cluster, there's a pharyngeal cluster that we helped identify. And this is contrasted, on the other hand, by synteny conservation on a much global scale on the level of chromosomes, so-called macrosynteny. And this is what originally was discovered as the Mala element. So when we were doing a lot of comparison on many animal genomes, already starting 10 years ago, we found surprising that many of those synthenies show some sort of conservation or retention from the last common ancestor of all animals, or Balaterians, somehow in the Cambrian or Ediacaran, so hundreds of million years ago, which was amazing because it indicates that those synthenies are highly preserved and still retain many of the species. But their function, as already mentioned, is largely unknown. So we would like to understand that, and my group in particular is focusing on emergence and syntenic transitions in specific group of animals. In particular, we focus on two very distantly related species. One is a cephalopod, Hawaiian bobtail squid, and another one is hydra, which have one thing in common: they have a very expanding genome compared to their closest relatives. And those expansions through transposons usually breaks up synteny, and we are trying to understand how much synteny has been broken up, how much novel synteny has been gained, etc. But we're also doing a lot of multi-species comparison. In particular, I have a team of really great, enthusiastic people who actually, one of them will be talking tomorrow about, Hannah will talk tomorrow about her work on cephalopods. So I will not talk about this today, but I would just like to mention some of the basic, more like comparative genomic theoretical analysis we are doing across many species to study how macrosynteny evolved. And so the two things that I would like to mention today is first the notion that macrosynteny or chromosomal scale synteny is preserved across many animals that we look at, but there are some peculiar exceptions, and I will talk about one of them. And also I would like to talk how these rearrangements in this ancestral macrosynthetic composition results in different properties of or emergence of novel microsyntheny, and how this may relate to novel regulation and things like that. So how do we look at macrosyntheny? So we usually, you can imagine this as comparing two genomes. And let's say if the genomes are like your genome, they're almost identical. If you align them together, you will see that there's a straight line. Everything is collinear. There's no changes that have occurred between the two genomes. But as the time progresses, population, species diverge. Then we have inversion deletions that accumulate. And this results in scrambling of gene order. But now the key thing here is that the scrambling is restricted, or at least that's the prediction of the model. The scrambling is restricted to chromosomal units because the recombination rate within the chromosomes is much higher than between the chromosomes. And so in the end, you end up with this kind of completely scrambled gene orders, but they're restricted to chromosomal boundaries. And so people have studied this for many years. And also one of the original papers in Amphioxus coordinate at the base of Retrogrades showed that it is also, even if you look at a very fragmented genome in context of scaffolds, you still find that you can recapitulate such a macrosynthetic pattern. You find those rectangles, so some people call them, which I really like, bags of genes, kind of still identifiable in this clustering algorithm. But the main question here still is, do those bags of genes correspond to actual chromosomes, or are they just some kind of more complex structures of synteny? And so this was impeded, of course, to answer this question was difficult without a chromosomal scale assembly. And we recently helped publish this Amphioxus chromosomal scale assembly using several methods, including the high-C method to stitch together scaffolds, using contact frequencies and 3D conformation of the genome, which is now becoming easier and easier. But of course, we verify this with a genetic map linkage. And what this allows us to see was the correspondence of those bags of genes that I showed you before to actual chromosomes in across animals, Balaterians, in particular Amphioxus. And this showed that whole chromosomes of Amphioxus actually correspond to those syntenic units. So you see here different chromosomes on the x-axis from Amphioxus, and they correspond to chromosomes of Scala that has been published by another group. And this is amazing because it really indicates that those are very ancestral Balaterian or even older linkage groups that are still preserved in very distantly related species groups. There have been some rearrangement and translocations that maybe if you see my pointer on the bottom side, but it is very rare, relatively rare. So this allowed us to reconstruct ancestral linkage groups. So in this case, ancestral coordinate linkage groups because we're looking at Amphioxus. And the majority of chromosomes in Amphioxus actually had almost perfect correspondence to those linkage groups. And so this allowed us then to study and the correspondence of this linkage group across animals. But the one that I would like to mention is one thing where all animals have this nice conservation of synteny. And if you look at Scala, but others, and everything is happy, everyone is happy. But then if you look at vertebrates, this situation becomes much more complex. There are many rearrangements, fusions, and a lot of work has been done to try to untangle this. So now with the chromosomal scale assembly of Amphioxus, we can try to address this kind of question. For example, we can say how many whole gene duplications there were and what really happened at the stem of the vertebrates. And so what we can find is that the ancestral linkage groups are largely retained also in vertebrates. You see here, you still see those bags of genes patterns, but they're multiplied. And usually you find 3 to 4 copies, which indicates that this is 2 rounds of whole gene duplications. But there are also some interesting cases of fusions of those ancestral linkage groups. For example, where you have per chromosome of GAR, in this case, you have combination of different Amphioxus chromosomes, ancestral linkage groups. And there are several types of those fusions that we can find. So one fusion is where you have a sharp boundary, like in this case, if you look at chromosome number 3, and this indicates a very recent fusion event of a chromosome. And then you have also more kind of mixed kind of, if you mix coffee and milk and then you have a mixture of colors, then this is also, you see it also in some of the chromosomes in chicken or gar, that some ancestral linkage groups, they fused some time ago, and this allowed them to mix over time. And then there are some chromosomes that are completely unfused, and those are, surprisingly, those are the microchromosomes of chicken and also in gar, which indicates that they are less derived compared to the macrochromosomal complement. But so some of the fusions are species specific and some of them are very ancient. And in the paper, we go at big lengths about describing and trying to plot this evolution. And we find that there's some fusions that were shared among all vertebrates, but they're specific to one round of whole gene applications. And in the interest of time, I just would like to refer to you to this paper if you're interested in vertebrate chromosomal scale evolution. But for the interest of this talk, I just want to mention that there were these very significant fusions that were not observed in any other animals. And what this means is that those chromosomal scale fusions, they brought together many genes that were previously unlinked. They were on different chromosomes together. And so the question that we would like to address is whether this has any impact on regulation or microsynteny in that sense. And so we have been doing this project actually not specifically for vertebrates, but across many species in parallel, where we try to evaluate our understanding of microsynteny evolution across all animals. But now I would like to mention mostly about something about the vertebrates. And so this re-evaluation of the microsynteny evolution with many more genomes really provides some very interesting insights in the dynamics of the small cluster evolution. For example, we find that many of the microsyntenic units originated in the Balatyrian animals. So some of those that were conserved already in the metazoan ancestor, but the majority of them actually originated, and you find them for the first time either in the Cnidarian Balatyrian ancestor or in the Balatyrian less common ancestor. And those are still largely preserved in many animal clades, except when you actually go to more complicated scenarios like vertebrates. So we want to understand what's really going on in terms of the organization of those microsyntenic clusters, comparing Balatyrian animals, invertebrate Balatyrian animals with vertebrates. And then we find that interesting insight into the scaling regimes of those synteny blocks. In particular, we find that some of the synthenies are much closely located together. The genes are more closely located, and those we call high density synthenies. And then we also find synthenies that are more loosely packed. Those we call low density synthenies, compared to randomization of the genomes that we performed. More interesting is that among all the synthenies that we look at, we find that in most invertebrate genomes, the majority of microsyntenic linkage groups are in these high density regimes, whereas only the minority are in the low density regimes. Whereas when we look at vertebrates, we find the opposite case, that the majority of microsyntenies that we find are low density, they are much expanded, and only few are high density. And this is also shown here on this kind of more complicated slide where we look at correspondence in the syntenic regimes between different species. And we find that in vertebrate uterostomes, they are largely high density as in the top right corner of this plot. But then vertebrates are completely depleted in this top right corner high density regimes. Instead, they either lost many of those linkages or they transition them to low density regimes. This basically, what it's saying is that you have more intergenic distances in those linkages in vertebrates than you expect by chance if you just look at the vertebrate genome. And this may be also corroborating some of the results that were published recently by Ferdinand Malitas et al. That showing, for example, you have many more regulatory sequences in vertebrate genes or around vertebrate genes. And so we kind of see this as a statistical support for that, just from looking from the genomics perspective. But there's one particularly interesting exception, and this is the Hox cluster, which many people have studied. In fact, the Hox cluster is the only cluster that is high density in vertebrates and low density in invertebrates. It's the opposite trend of the general trend, or it's exception to the rule. You see here also the genes in the Hox cluster, as people have shown, also are tightly packed, whereas genes of Hox genes in invertebrates are completely or they are more spaced apart. And this scales very well with the genome sizes of all genomes that we looked at. So this is not a new finding. People have found this very nicely also before. But what I think we can contribute to this is that we can say that we can distinguish between the two different regimes. And we can say that one is low density statistically and the other one is high density. And then we also can see that generally the syntenies have different topological organization. From the genome, for example, looking at the high C density map and regulation as has been published by some colleagues. Hawk's cluster has prominently Hawk's D class has 2 TAGs, whereas topologically associated domains, whereas the invertebrate Hawk's cluster has presumably only 1 topologically associated domain. So the contacts are mostly within the cluster than to the outside of the cluster. And this we find to be also in general a feature of those high density versus low density syntenies. So in the conclusion, we see that while both syntenic regimes in microsynteny co-occur in animal genomes, and while one regime is more predominant in one animal group than another, they likely can be used as a predictors of different topological organization. So to come back to the big question is why are animal genomes organized the way they are? I think we see statistical evidence that there is some sort of pressure to maintain particular topological organization. There's evidence for co-regulation, also evidence for co-expression of some of those at least, and this we published in different paper. And so some people, when they saw those results, they proposed that this is maybe some sort of a symphony of synteny concept, because you have such complex regimes and different co-regulation, etc. But of course, we have to know that this symphony is different depending on which animal group you look at. So in your average, let's say, metazoan species, you will have very nice conservation of macrosynteny, high density microsyntenies, whereas some species just completely take this apart. Invertebrates, as I've shown you, they switch the regime, they fuse a lot of macrosynthenic regimes. And also some other species as well. And so if you would like to listen to learn about the other exceptions in cephalopods, then you should listen to Hanna Schmidbauer, who will talk about them tomorrow. So with this, I would like to thank especially my group, who really are outstanding, and I'm very happy that they can tolerate me. And I would like to really thank also our collaborators on different projects, and special thanks to my former supervisor, Dan Roxa, and also all the funding sources. And also thank you for your attention, and I'm happy to take questions.
Thank you, Oleg. We have time for a couple of questions. So David Ems is asking through Slack, do you think the synteny conservation is giving a measure of something more biologically important that is being conserved rather than just the synteny itself? And if so, do you have any idea what the biologically important properties might be and what further data we need to investigate which properties?
Mm-hmm. Yeah, so I think now we have some evidence that this is synthenies that, or genes that live in syntenies, they also tend to co-express in some of the species, that they, the closer they are, they have a different topology or organization of the, maybe the regulation than genes that are most further spaced apart. And so I think there is something functional to it. The question now, is this the same functional topological organization, 3D regulation of the genome for microsynteny as well as for macrosynteny, or these are two different things? Is maybe macrosynteny is just a property of how chromosomes evolve, because you cannot just fuse randomly chromosomes. It's much more difficult process, and it requires a lot of population support, population genetic support. Whereas if you have more or less genes that move within the chromosome, it's more likely to obtain it, and this can quickly change a regulation mode. Yeah, and what data we need to more species, more more of everything, expression, high C, and attack, and whatever. Yeah.
Okay, and perhaps the last question by Kristina Gagalova. Are the genomes that you are comparing the same contiguity? In that case, how do you cope with those differences?
Yeah, That's a very good question. So even if the genomes are assembled to chromosomal scale, they are not of the same quality because it is, you know, there are different methods used. Some people verify the assemblies more, some people less. And how do we use this? How do we approach this? We usually bootstrap the data. We run different simulations. It depends on the question a lot. Yeah. For example, people talked before about orthology, so we try different orthology methods. And generally, we make predictions, and then we see if the species, new species that has been sequenced, confirms those or not. And this kind of evolutionary conservation is a significant verification of our predictions.
Okay, thank you, Oleg, for this very nice talk. I think we should move on with the next speaker.
Thank you very much, Oleg. He will be able to reply to your questions. I've seen there are many here afterwards, written replies.